Now, we talked about in the previous section how scientists engineer DNA sequences that they would like to put into plant cells. But we did not talk about how the DNA can get into plant cells. Historically, most of the plants that were genetically modified were done so using a technology called biolistics, otherwise referred to as the gene gun. The basis of this technology is essentially like a gun. Initially, gunpowder was used, but more recently, for safety reasons, pressurized noble gases like helium are used. The DNA is loaded onto a gold plate just above a piece of plant tissue. This pressurized gas is then loaded into a chamber and released to spray gold particles with DNA attached onto the plant tissue. Many of the cells in the plant tissue will be damaged in this process, but some of them will end up having DNA enter the cells and not completely destroy the cells. The cells can then heal, and the new DNA that's been inserted into them or shot into the cells then ends up becoming incorporated into the DNA itself. This process is very primitive and not very efficient, but it does work. And because it was so easy to manufacture, scientists were using that for a long time. However, there is a different technology that can be used in some organisms to more efficiently engineer or insert recombinant DNA into a cell. And that is using a bacterial cell called Agrobacterium tumefaciens. Agrobacterium is so named, agro from agriculture, and bacterium because it's a bacterial cell. So this is a bacterial cell that loves to live in and around the plants that we grow for food. Now, it just so happens that for millions of years, this plant pathogen has evolved the ability to insert some of its DNA into a plant cell. It naturally does this in the soil for millions of years prior to human existence. So in fact, Agrobacterium, you could say, is the first genetic engineer. It evolved the ability to insert its DNA into the plant cells because that DNA would produce Agrobacterium food. So Agrobacterium was essentially using plants to make food for it. What scientists have done is they've removed the genes to make Agrobacterium food and inserted the recombinant DNA instead. And now what the genetic engineers can do is put that Agrobacterium cell onto any different type of food plant and get the Agrobacterium cell to insert the recombinant DNA into the plant cells itself. And because this is a natural process that evolved over millions of years, it is extremely efficient and does this work very well. However, because this is a newer technology and not as easy to perform, biolistics, or the gene gun, was used to manufacture most of the GMOs we have today. Finally, once you engineer the cells, the plant cells, you can grow up the plant cells into cell clumps, just on petri dishes. Now these are sort of amorphous clumps of plant tissue. There's no leaves or roots, as you can see here. But in fact, it's really easy to generate leaves or roots from these clumps of plant tissue. All you have to do is add a few plant hormones and you can get some roots to grow and some shoots to grow. You can then allow these to grow a little bit longer on a petri plate and eventually plant them into the soil. And once you do that, you can have a transformed or genetically engineered plant. These are your GMOs. Now, what kind of GMOs do we have today? Well, we actually have been growing GMOs for almost 40 years. The first one that was produced for commercial purposes was in 1993, and it was referred to as the flavor saver tomato. Spelled a little funky without the E's, the flavor saver tomato was a scientific success but a commercial failure. The difference between the two is that the non-genetically modified tomato would rot and go bad on the shelf within a few days. This is why most people will stick their tomatoes in the refrigerator, even though this does alter the flavor of the tomato itself. Scientists wanted to generate a tomato that could be shelf stable and not rot or go bad on the shelf, thus also retaining its flavor for weeks. And that's what the flavor saver tomato was about. However, it did take some time and effort for scientists to generate this. And so as a result, because it was so expensive to manufacture, it was expensive to sell. And consumers didn't see the point in purchasing a tomato for $5 that they could use in 3 weeks when they could just eat a tomato this week for a lot less. So even though it was scientific success, it was a commercial failure and nobody ended up buying it. Those genetically modified tomatoes, the flavor saver types, and any other type that was genetically engineered, are no longer for sale commercially. In fact, none of the tomatoes grown in the United States today are genetically modified. That may change in the future, but for right now, we can say that there are no tomatoes available for sale. Instead, we have other plants. Soybeans are a really common one that's genetically modified, and so is corn. Here we have a genetically modified corn and a non-genetically modified corn. And anybody who thinks that genetically modified foods are scary would probably think that the GMO version is this bottom one. But in fact, that's the non-genetically modified version. It's this top one that's genetically modified. Because the corn here that you see was genetically modified to resist pests. It's BT corn. And BT corn is a lot less susceptible to pest infestation and disease than its non-genetically modified relative. Now, as I mentioned, most corn and most soybean are genetically modified to either be Roundup Ready or contain the pesticide BT. Cotton is also widely genetically modified. Over half of cotton is genetically modified. Canola for canola oil. Alfalfa for animal food. Sugar beets used to produce sugar. Most people think that their sugar comes from sugarcane, but in fact that's not true. If you buy any baked good, any breads, cookies, crackers at restaurants, bakeries, Starbucks, or any place like that, nearly every time their sugar is going to come from sugar beets, genetically modified sugar beets, and not sugarcane. The sugar from sugarcane is actually more expensive. There's also genetically modified papaya from Hawaii. About half of the papaya from Hawaii is genetically modified. There's a crookneck squash that's genetically modified to resist viruses. And the papaya from Hawaii resists viruses. Apples have been genetically modified so that they don't turn brown when you cut into them. These you really only find in salad bars or McDonald's Happy Meals, where pre-cut apples would be unappealing if they were brown. There's also potato that is genetically modified. And finally, there's salmon. Salmon was the first and so far only animal to be genetically modified and sold as a GMO. It's genetically modified so that it's sold at 1 year old instead of 2 year old because it grows twice as fast. So half of the food is needed to produce it, and it produces only half the waste. It's in fact extremely environmentally friendly as far as farmed salmon goes. However, still to this day, the most common genetically modified products are corn and soybean. But don't think that these products, corn and soybean, are sold to you as corn cob or kernel corn or soybeans that you would have with sushi. The genetically modified corn that's produced in the United States, and there's a lot of it, is either used to produce corn syrup or used for animal feed. Soybeans, likewise, are not eaten in the genetically modified version as a pure plant. Instead, soybeans that are genetically modified are used to harvest partially hydrogenated soybean oil, and that is placed into processed foods. So it's processed foods primarily that will contain these genetically modified ingredients. Now, in our last section, we're going to talk briefly about concerns over GMOs.